today, the 20th of May, the Clinical Trials Day, and it's actually on that occasion that we have had the idea of hosting this event around clinical trials, because of the Clinical Trials Day, and it became quite quickly clear what topic around clinical trials we should cover this year. And this is, of course, the vaccine development, with all that we hear nowadays around the pandemic and the vaccines that have been developed. And there's a lot of people having a lot of questions around that development, how it is possible to develop them so quickly, where normally it takes much longer. There's also like people that are concerned about the safety of vaccines. There's people that don't want to be vaccinated. So what we thought is we are going to organize this session for a very large audience, and this is also the case. There is people that are experts in vaccine development. They work in companies developing vaccines, but there's also people, lay people, that are just curious about clinical research and vaccine development that don't know anything about it. So this is a challenge that we have to manage expectations from both groups. But we're trying to meet that as good as possible. So before we jump into the presentation, I just wanted to make you aware that we are going to record the session. So if anyone would have an objection against that, please feel free to raise your concern in the chat, or leave the meeting if you don't want to be registered. When we register, the names will not appear. It will only be the presenter and the slides that appear. Having said that, as you may have seen in the first slide, the event that we host today is kindly hosted by ECCRT, the European for Central Clinical Research Training, which is a professional training organization. And what we are passionate about is to develop the people to do better clinical research in a better way, in a more effective way, for the benefit of patients. That's what makes us getting up in the morning every day, enthusiastic to organize our courses, our information events, everything that we do. And we do, as you can see on the slide, quite a lot. So we have over 100 different courses, all related to clinical research. So how are drugs developed? Not only drugs, but also medical devices and vaccines. So there is quite a lot of things happening. We have more than 50 trainers, and we have trained more than 14,000 people in almost all countries in the world. And so we're happy to also contribute to the general knowledge about clinical research to the wider community. Having said that, let me jump into the agenda for today, and it's like a quite nice agenda, again, around surrounding clinical research in developing vaccines. We'll have three parts in the today's session. I will give a very short introduction about clinical research, what it is, for the people that have no clue what it is. So it's going to be very foundational, very short for those people. But then the main part of the discussion today is going to be around clinical development of vaccines specifically, and how important this is in general, and also specifically how it has been done for the COVID-19 vaccines, and how it has been the people have been able to accelerate the development, and how this has been working. The last part, so part 1 and 2 will be in total around about 1 hour, and then we'll have half an hour approximately for question and answers, because we want to give also you the floor and the opportunity to ask and raise your questions. The way we are going to ask you to raise questions is to put your questions in the chat. You can reach the chat on top of the screen in Teams, this little call out icon. If you click there, you see the chat, and yeah, you can put your questions. So we will be monitoring those questions during the session, and then we'll address your questions at the end. If you really want to speak up in the question and answer session, you can use the raise function. And Maybe one more house rule to announce here. If I can please ask you to all put yourself on mute if you're not done so, just to avoid that there's too much background noise. So if that will be possible, that would be very nice. So feel free to think of your questions, write them in the chat, and then if you want to speak up later on in the Q&A, you can use the raise hand function, then we'll give you the opportunity to speak. So we have quite some people attending, but we still want to make this an interactive session for you. So looking at the agenda for today that you see on the slide now, I'm very pleased to introduce our main speaker for today, which is going to be Professor Isabelle Leroux-Rouls. And for many people, she is not unknown, and she's very active in the field, as she's heading the department of the Center of Vaccinology of the University Ghent at CEVAC for quite a while, already since 2017, and actually been involved in CEVAC since many more years. Professor Leroux-Rouls is a clinical microbiologist and an infection control physician in the same hospital, in the University Hospital in Ghent. And in that capacity, she has done a lot of clinical trials with vaccines, especially also with the COVID-19 vaccines that now recently have been taken out, and kept her very busy and kept her nights very short, as she says herself. Before I give the word to Professor Leroux, I'm going to cover that first introduction part, and maybe because I didn't introduce myself properly for the people that don't know me. My name is Benedict van Nieuwenhoven, and I'm actually the Managing Director of the European Centre for Clinical Research Training, on the one hand, and on the other hand, I am also a professor at the Faculty of Pharmaceutical Sciences, teaching a course, Management of Clinical Research. And well, actually my whole career, I have been involved in clinical research, and I'm now quite passionate to be able to share that knowledge to a broader audience, and I'm very happy to do this today as well. All right, so before we jump into the content, as I mentioned, we're going to make this session quite interactive. So I am also going to call upon your input here. So we are going to organize a number of polls during the session, and these polls are going to appear on your screen, and I'm going to launch one right now. So just to get a better understanding of who is in the audience, I'm going to launch this survey right now. If all goes well, you should be able to see the question coming up. If it's not coming up on your screen, it might appear in the chat. So feel free to answer the question to the best possible answer. So if you are a citizen without any scientific knowledge, you select the first box. If you're a scientist but not involved in clinical research, you can do that. Or if you're part of a vaccine company, that's the opportunity to check that box, or another pharma company, or a CRO consultancy, or if you are on an investigational site. So doing clinical research in a hospital or in a clinical unit or alike. So you have the opportunity now to answer those questions, and I see that a lot of people have already answered. Feel free to still add your answer. I've seen that 33 people have voted already, so I'm going to give it a little bit more time for all of you to vote. So I see that indeed 1 out of 4 attending here today is working in a company developing vaccines. So I'm sure you are the experts. So probably the first part is not going to be saying a lot to you, but still there is also the other people, and there's also like 10% like citizens that just are interested in how these vaccines were developed without that scientific knowledge. So I still I think it's important that we also like cover the basics for the people. So thank you for voting on that one. I also have like maybe a provocative poll for you. Another one is about vaccination actually, and whether you're already vaccinated or not. So I'm also going to allow you some time to answer that question. So if you have been vaccinated first injection or completely, or it's being planned, or you have like a schedule already, or nothing is planned yet, then you're probably in a younger age group nowadays, or you are belonging to the people that don't want to be vaccinated. So I'm curious to see the results on that one. So interesting to see there's 1 out of 5 that has already been completely vaccinated. More or less, there is like the same amount with the first injection, 1/5. 15% has a planned vaccination going on, and 40% has not yet received any invitation for vaccination, and there's 8% of the people that do not want to be vaccinated. I'll be interested also to hear their voices and to hear what their concerns are with the vaccination, and I'm sure we'll also be able to provide you with some information or relieve some of the concerns that you may have. All right. As time progresses, let me jump into the first slide of the content, and first slide is actually about the Clinical Trials Day. Just because people may ask, what is the Clinical Trials Day? And why is it the 20th of May? This is a global day, so this is celebrated over the entire world. And the reason is that actually on the May the 20th, James Lind started the very first ever controlled clinical trial. So basically that we consider that as the start of the clinical research. That's quite some time ago. This is in the year 1747 actually, so it's quite a while. So he was doing a scurvy study where he divided a number of people in groups, which made it a controlled trial, so that you don't have only one group of people with an experimental treatment, but you divide it and use control groups like placebo-controlled, or you have an equivalent treatment, for instance, to compare it with. So that's the reason why 20th of May is the Clinical Trials Day. And we also use this actually to recognize people that have made clinical research possible. In the first place, the clinical research professionals doing clinical research, and all the investigators, study coordinators, the companies doing clinical research and making it possible that new therapies are being developed. And that's quite important that this continues. But equally important, also the people that participate in that clinical research, because this is, well, actually without them, there would not be clinical research. How much effort we would even try to do without the people that volunteer to participate, we would not have any clinical research. And that's also then the secondary reason for that Clinical Trials Day, is to create that awareness. And that's also why we organize this event, just to make clinical research more commonly known. Most of the people still think that you are like a guinea pig if you participate in clinical research, but it's more than that. As most of you know, in the meantime, you're really contributing to advancing knowledge and advancing new therapies for better treatment or preventing of treatment of diseases for the future. And very often you get more access, early access, by participating in clinical trials, but actually the more important thing is that there's future cures for future patients, and I think that's where the importance comes from. Now, I've talked about clinical research already quite a bit, so let me zoom in on what it is precisely, clinical research, because this is only part of the whole development of a new drug. And the slide that you see on the screen now is the entire drug development cycle. And I'm speaking here drugs, that there's also other, the regular drugs. Vaccines are slightly different, and we'll come back to that later. but overall it's comparable. It's consisting of 5 parts, as you see. So you have in the first place the drug research, where this is the lab work that is done in the lab, testing and looking for potential compounds or products or molecules that could be further developed. And you see also on the bottom of the slide the duration of this whole timeline and the amount of compounds that are being tested. So on the slides, it's on average 10,000 compounds that are initially tested to actually get out one drug or vaccine on the market. It's actually a lot that is being tested before one comes out on the market. So a lot of compounds are being dropped along the way. So drug research, the lab work, is the first part. Second part is the preclinical work, where there's still lab work and animal research. So that's the studies that are being done on animals to get information on the behavior in the living species before we go into human beings, because that's precisely the point where clinical research starts. When the test compound is administered to a human being, then we speak about clinical research, clinical trials. Clinical trials are typically divided in 4 phases. The first 3 phases are prior to the launch, and the phase 4, the last phase, is after the evaluation and the approval. Now, those phases, just for the people that have no understanding of how clinical research is being done, as mentioned, is 4 phases. The first phase being the first time it's really administered to human beings. So people are very careful there, and overall, in general, it's done on healthy volunteers. And the purpose is to investigate how the new compound is behaving in the human body, and how does it affect the vital organs, for instance, and how the body behaves. So it's mainly safety, and see what happens with the compound in a human being. And that's why it's done on normally on healthy volunteers, with few exceptions. Sometimes there's also like phase 1 trials on patients in oncology, but mainly it's healthy volunteers. We talk about patients when we move to phase 2, where then you move into a patient population that has the targeted disease. And you do this on a very small group, because again, you don't want to expose the test compound to a too big group in the first place. And you also want to narrow the group down to really the people that have that disease only. So you want to exclude people with other diseases in parallel, or that have other conditions that could have an impact on how the drug is behaving. So you want to make as a clean population. Also, not a very high group of patients is being studied. The bigger groups of patients are being tested in phase 3. That's where you move then to a situation more close to real life. Not necessarily completely real life, as we'll see later, but at least there's a larger population being included in those trials. That patients that have also other comorbidities, other diseases, or that do take other medication, for instance, that also can have an effect on the drug to be tested. And because of course you also want to investigate that, you want to research if another disease could have an impact on the efficacy and the safety of your test compound. Talking about efficacy and safety, as you can imagine, in phase 1, it's only safety purely. In phase 2, still safety is the main criteria to be tested. However, there you're also going to already get a first indication about effectiveness. The main study phase for proving that the drug works and is effective is phase 3. And as you saw in the previous slide, once phase 3 is successfully completed and you have been able to demonstrate that your drug is working and it is safe, then the the producer of or the sponsor is going to submit for a marketing authorization, and then the authorities, the regulatory authorities, are going to review your dossier with all the research that has been done, not only the clinical, but also the preclinical and the lab research. And they're going to evaluate that, and we'll talk about that later. Isabel will focus on that, how that is going for vaccines. And so after the evaluation, you will get a marketing authorization or not. So when you get your marketing authorization, very often condition will be that you also continue to monitor your drug, okay? Because when it's put on the market, immediately you have a much larger audience and a much larger population that is going to be treated with your drug at that time, which also then explains as to why sometimes side effects occur only after the trials, because actually the frequency of that adverse event or that side effect might be so low that you actually don't even get to see it in the clinical trials phase 1 to 3, because you're not including enough patients to see it. Even in an adverse event, in the side effect only occurs in 1 out of 100,000 persons, for instance, you might not even get to see one patient experiencing that side effect. All right, and that's why this phase 4 trials to like continue to monitor the drug and to evaluate it in real life conditions. Right, and then let me, like, with that slide, I'd like to then slowly transition to Professor Leroux's, and let me show you that slide here on vaccine development and the timeline. Timeline is comparable. Of course, the biggest difference developing vaccines versus the classical drugs, the medication to treat a disease, is the fact that a vaccine clinical study is not carried out on patients. It's always done on healthy volunteers, as you can see in the slide. And that has consequences, of course, because you need to find those healthy volunteers that have not been immune compromised, etc., etc. But I'm sure that Isabel will talk more about that moving forward. Equally and similar to what you saw in the drug development timeline, it takes many years to develop a drug. A drug can take 12 to up to 15 years, depending on the class of product. If it's a completely new class, it may take longer. If it's a drug that has a similar drug already on the market, it may be shorter. Also, as you can see, it can take several years to bring a vaccine on the market. And just to illustrate you, my last slide here for now shows you a number of vaccines that have been developed for a number of infectious diseases, with how long it took to develop them. And as you can see, for certain vaccines, it has taken over 10 years, up to 20 or even 30 years. So it is obvious that people have the question now, why the COVID vaccines were developed in such a short time, as in 1 year time, basically, they have been brought on the market. And I'm sure that indeed there is concern about that. And I'm now happy to transition to Isabel, who is going to also like deal with that and how it's been possible to reduce those timelines. So Isabel, if you are ready to take over from me, happy to give you the stage.
Thank you very much, Benedict. And good afternoon, everybody. Thank you for the invitation and the introduction. And I am indeed going to take over here. So the importance of clinical research in developing new vaccines, and in particular COVID-19 vaccines. And as a principal investigator of 6 COVID-19 vaccine trials in the meanwhile, I'm also happy to share with you some personal experience, myself and my team members, of which some are also joining this meeting. So I've a quick overview on the structure of my talk. So very briefly, vaccines, I think we all know they are a global health success story, but there still are some challenges. And new vaccines are needed, not only against COVID-19. And then specifically focusing on COVID-19 vaccine development, this accelerated development process. How did it go? Why did it go so fast? And to reassure people in particular. And then being the International Clinical Trials Day, what have clinical trials or clinical research contributed? What have we learned from the trials that have been conducted? And what will the ongoing, but because still many trials are still ongoing and future trials are planned, what will we know from these studies? And what will real life experience tell us? Because not all scientific questions can be evaluated in clinical trials. And for some information, we have to rely on epidemiological data, virological data, and real life experience. And then, of course, I'm going to conclude and leave sufficient time for interaction, discussion, and all the questions you might have. I think we could spend a day talking about the successes of vaccines, but just summarized in one slide, vaccines are considered together with clean water and sanitation and antibiotics as one of the greatest public health achievements. WHO estimates that around 2 to 3 million deaths per year are averted owing to vaccination. Vaccination has eradicated smallpox, and we are quite close to the eradication of polio. They have reduced global child mortality rates and prevented countless birth defects, and vaccines will play and are already playing a crucial role in controlling the ongoing SARS-CoV-2 or COVID-19 pandemic. But there are, of course, challenges as well, and you can have the best vaccines in the world if people don't want them. If they don't have them injected into their arm, they are of no use. And already in 2019, WHO declared vaccine hesitancy as one of the 10 threats to global health, together with pandemic, influenza pandemic, and with high threat pathogens. I think the current pandemic nicely demonstrates we have actually, well, this situation, 3 of these threats, we are living them right now. It did not become an influenza pandemic, but a coronavirus pandemic. But otherwise, we are living these threats. And vaccines, they are very important in a way that they do not only provide individual protection. That's why this vaccine hesitancy is such an important threat. It's not about individual protection, but it's a fact that if a lot of individuals refuse to be vaccinated, that this poses a threat to the global public health, because vaccines have this additional feature that by vaccinating sufficient people, you get indirect protection of those that cannot be vaccinated, which we all know called herd immunity or group protection, group immunity. It's a very busy slide, and I'm not going to go into detail, but it's just to give you an overview of how many vaccines are already on the market, licensed and used. More than 30 vaccines are vaccine preventable today. Each of the colored dots actually shows the year in which this specific vaccine became licensed, and the color indicates the technology or the platform that was used to develop this vaccine. And then the timelines below show you that actually all of the technologies, with the exception of variolation against smallpox, that has been that's a technique that is not in use anymore since many years, but all the other technologies are still being used, from the earliest, the live attenuated and inactivated vaccines, over the toxoid and polysaccharide vaccines, up to the vaccines that only use a small part of the pathogen of the virus, which we call the split or subunit vaccines, over various types. And I've added myself SARS-CoV-2 or COVID-19 with 2 new colors, the pinkish one and the orange one, because it actually, this pandemic has led to the acceleration of the development of novel platforms that were, of course, not discovered just now. No, these technologies have been in development already many years, and there were various clinical trials ongoing. But this pandemic has actually been an opportunity to accelerate the development. So it's the mRNA and the adenoviral vector, and I want to be correct in a way that the first adenoviral vector was actually already licensed last year in May. in 2020, and that was an Ebola vaccine developed by Johnson & Johnson and used in Africa. SARS-CoV-2 vaccines, there are also inactivated vaccines that are being used, but not licensed in Europe yet, but in other parts of the world. And we expect quite a lot of protein vaccines or subunit vaccines to be licensed in the coming months. So we already have quite a lot of vaccines, but there's still many vaccines we need. And what are the challenges for vaccine development? We could categorize them or put them in three categories. First of all, there's challenging pathogens. Those are the pathogens which cause a lot of disease, a lot of deaths, but against which we don't have a vaccine today. Examples are HIV, tuberculosis, RSV, and so on. I think we could also say that a SARS-CoV-2 virus is a challenging pathogen because of its mutations and the variants of concern it causes. Then there's challenging populations. We know that vaccines the licensed vaccines are working well in healthy individuals, but very often the most vulnerable people, older adults, immunocompromised persons, do not respond that well and might need a different approach. And then thirdly, there's challenging circumstances. And again, I think this current pandemic has put us actually in these three situations. It's a challenging pathogen. We know that older people and immunocompromised people are at highest risk and respond have the lowest response to vaccination. And then we have the specific circumstances of having a worldwide pandemic affecting our healthcare system, all of our society, and with the whole world being immunologically naive. So we need a huge number of vaccines and the production capacity is limited. So there's various strategies to overcome these challenges, and all of these strategies from new antigens, delivery strategies, adjuvants are being explored, are being used to develop new vaccines against COVID, but also against various of the infectious diseases listed here. Well, so we need a lot of innovation, and I already, I don't know from everybody where you're exactly located, but I dare to think that we have a very international audience, but also quite some people from Europe and also from Belgium. So a lot of the innovation with regards to vaccine development, vaccine production is taking place in Europe. And these are data I found on the website of Vaccines Europe from 2019, showing that every year 1.7 billion vaccines are produced in Europe. So counting for a large proportion of the world's capacity. And we have already seen that this production capacity, although it is high, it is not high enough when a pandemic strikes us. Vaccines are produced in 11 European countries across various production sites, and there are also research sites, research institutions that are leading discovery of next generation vaccines, addressing unmet medical needs and support to face emergency situations. I'm not a chauvinist, not at all, and Belgians are in general considered to be humble people, but still I want to present some Belgium figures at the European Centre for Clinical Research Training. Organizing this event is located in Brussels, and the Centre for Vaccinology is located in Ghent, so two Belgian cities. So just briefly some data on Europe and Belgium from the pre-COVID-19 era. I was unable to find an update, but showing you the number of clinical vaccine trials that is being conducted in Europe every year. It's between 60 and 130 trials per year, and you see a decreasing trend in the orange bars, which could be considered a bit worrisome. But you see the proportion of studies being conducted in Belgium, the blue bars, is relatively stable, and it accounts for between 10 and 20% of the clinical trials. So we're quite proud of that because we're a very small country. Our population only accounts for 2.5% of all Europeans. So clinical research is an important activity in our country, and it is linked to the fact that we have some large vaccine manufacturers which are based in our country, in Belgium, and the fact that there are several dedicated vaccine evaluation centers. And then thirdly, our federal agency for medicines and health products, who provided this slide, they have vaccines as their focus, as one of their main spear points. The majority of studies being conducted in Belgium are commercial, industry sponsored, and mainly phase 1 and 2. So for phase 3 trials, companies tend to go to other countries where the costs are lower. Although for the phase 3 COVID trials, quite a lot of activity was ongoing and is still ongoing in Belgium. So we are quite proud, and I apologize for those among you who are not Dutch speaking, but this newspaper article of last week says that Belgium is considered as a vaccine country and has its position in the world for the various reasons I just mentioned. So switching gears or diving into the COVID-19 situation, just a quick view on the daily updated COVID-19 dashboard from the Johns Hopkins University, showing us that we have already 164 million cases, reported cases, and almost 3.5 million deaths. As I said, reported cases and reported deaths, because we all know that this is an underestimation. A lot of cases go unnoticed, unreported. So the impact of this pandemic is much higher than what is shown here. In the meanwhile, vaccination rollout is gearing up, and 1.5 billion people have already, or 1.5 vaccine doses have already been administered. So some people have received 1 dose, others have received 2 doses, but it is only 15% approximately of the world population. So still a lot of people are susceptible, and as we know, various parts of the world are still in a very serious situation with regard to the circulation of the virus. India, for example, you can see it on the left-hand side, after the US, India is most affected by the pandemic, of course, based on registered numbers. I think the underreporting must be substantial in a country like India. On 11th of March 2020, when WHO declared the pandemic, the race for the vaccine, and how media tend to call it, has started, but actually it started before. We already knew that this virus emerged end of December, and scientists all over the world were already thinking about which target antigen to include in the vaccine. So the first clinical trial started already in April. So the race started, and it's based on previous experience with other coronaviruses. We knew that the spike antigen was going to be the main target. So vaccines, actually, what do they do? What is their aim? Inducing specific immunity against this virus and against the spike, and even more specifically against the tip of the spike. The tip of the spike is called RBD, or receptor binding domain. It is with this tip that the virus attaches to our cells via a receptor. So that is the primary aim, inducing antibodies against the spike protein that are able to neutralize the virus and prevent that the virus can infect our cells. But a good vaccine not only induces that type of antibodies, also induces other types of antibodies that can play a role in preventing infection and induce cellular immunity or cell-mediated immunity. The most important cells that play a role are T cells, helper T cells, killer T cells, and B cells. And these cells, after being activated, can turn into memory cells. And these cells are important upon a next dose of the vaccine or upon an encounter with the virus. They can be rapidly reactivated and ensure a so-called booster response. There's various ways of inducing that specific immunity, and actually 7 different technologies are being used and are depicted on the right-hand side. But it's actually more or less the same of what I've already shown you, going from the traditional the old-fashioned, if I could say so, technologies, the live attenuated, inactivated vaccines, over the ones that only just use a small part, being the protein, the spike protein. So these traditional technologies are being used, but the ones that were fastest in evaluating, in being licensed, are the ones here on the lower part of this figure, being the mRNA vaccines and the viral vectored vaccines. The one I have indicated in bold are the ones that have been licensed already in Europe by the European Medicines Agency. CureVac is also an mRNA vaccine from a German company. We expect results very soon, and we hope it can also be licensed. And we know that the Russians have also submitted their file to EMA, so it's possible that this vaccine becomes available in Europe. I forgot to mention the exact correlative protection. What do I mean with that? Is that today we know that all of these components depicted here are important, but we do not know exactly what the minimum titer or quantity is of these antibodies in order to prevent infection. We're making a lot of progress. Scientific world is making a lot of progress based on challenge models in non-human primates, based on the follow-up of individuals and the reinfection rates. So various types of studies are helping to get to know what we actually need, because that would be very helpful. Also, after vaccination, some people are having their antibodies tested, but right now we're in a position where we cannot say to somebody, well, you can sleep on both ears. You have this antibody titer is sufficient to be protected or is not sufficient to be protected. This is something which we would like to know and we would like to have and hope to have in the near future. So I also introduced a poll question just to keep you awake and alert in this afternoon. It's maybe a little bit of a silly one, but anyway, just to see if you have any idea at all. It's based on a New York Times Corona vaccine tracker. How many SARS-CoV-2 or COVID-19 vaccines are currently in development, meaning phase 1, phase 2, and phase 3, so being tested in humans. Not the ones, of course, already in use. Would you have any idea? You can indicate the box that matches your guess. I'll give you some time. Okay, yes. The numbers are not that different from each other, so it's not that it's this important knowledge, not at all. Yeah, I see. 42% has indicated option C, which is indeed the correct answer. It's quite impressive to see. This is the coronavirus. It's a very interesting, it's for a very general public, but a very good source of information on the vaccines. A timeline of all the milestones that are being reached, how these vaccines work, and they also keep track of all the trials that are ongoing. And indeed, 113 vaccines are in phase 1, phase 2, or phase 3. And if you add up the vaccines that have been authorized for early or limited use and the ones that have been approved for full use, you end up with 128 vaccines. So that is amazing. 15 have been licensed or partially licensed. So 15 vaccines in about 15 months is incredible, is impressive. And it is for large part thanks to clinical research, to networks, to the effort of companies, to the budgets that have been made available. 4 vaccines have abandoned or have been abandoned. Their development has been stopped after disappointing results in the clinical trials. So that's 4 out of 20 approximately. So that's 20%, which is not that high, I think, if you compare it to the success rates overall. I think it's quite high success rates. of course, that number of abandoned vaccines will obviously increase with time as well. The development and approval stages have already been summarized by Benedict, and I will come back to that later when we talk about the acceleration of the process. And this has already also been alluded to by Benedict in his first talk, is what is typical for a clinical vaccine trial. There's many differences as compared to drug trials, but indeed, as you already said, Benedict, an important difference is the fact that phase 1, 2, and 3 in vaccine trials are done in healthy volunteers. Phase 1 is done in young adults, 18 to 40, 18 to 50 years. And from phase 2 onwards, we can start including older age groups or younger age groups. Mostly the vaccine is fully evaluated in adults, and once it is approved, then the evaluation in adolescents and in children is started. Of course, as you progress into phase 2 and phase 3, the criteria to participate become less stringent. So people with chronic diseases can participate. It's really also a requirement. It was a requirement from FDA, EMA, that a substantial proportion, preferably 25% of the participants, was 65 plus or had a chronic disease, a well-controlled chronic disease. But important risk groups are not included in the clinical trials. For example, the residents of long-term care facilities who are very old and very frail individuals were not included, although they were very affected by the pandemic. People with immune suppressive conditions and immunosuppressive medication, with the exception of well-controlled HIV infection, these individuals were also not included in the clinical trials. This has as a consequence that the risk-benefit ratio of a vaccine is considered completely different compared to a drug or medication, in particular if it's a drug that treats a disease that is potentially life-threatening. So in case of a vaccine, you administer it to large numbers of healthy people who might not even get this infection, who might not even become ill through COVID-19. So that means that the advantages, the benefits of the vaccine must always largely outweigh the risks, the side effects that the vaccine causes. The duration of a vaccine trial, in particular, if you compare it to phase 1 drug trials, is also much longer, between 6 months until 2 years. That is standard. Whereas a phase 1 drug trial, which focuses on pharmacokinetics, pharmacodynamics, often just takes a couple of days to weeks. The reason for this long duration is to look primarily at the persistence of immunity or protection, to see how the antibody titers or the protection decrease with time. And that takes, of course, time to follow up. A lot of people think that a trial takes that long time to follow up on the safety of a vaccine because we expect side effects after 6 months or a year or 2 years. Of course, when a study takes that long, we also document the safety of the vaccine, but we don't expect any side effects long time after vaccination. We expect side effects the first 6 weeks, and that is based on the many experience there is with the development of all of these vaccines. You expect side effects the first 6 weeks after vaccination when you induce or stimulate the immune system. After that period of time, it becomes very unlikely that you will see any side effects. And then thirdly, the vaccine trial size. Of course, I will not generalize that. There are certainly also drug trials that are quite large, but in general, vaccine trials, in particular phase 3 trials, are very large studies. But you cannot say that there is just one standard size for a phase 3 vaccine trial. It depends from vaccine to vaccine. And just to give 3 examples, meningococcal vaccine trials phase 3 are generally quite small, smaller than 10,000 individuals, because for meningococcal vaccines, it is sufficient to provide antibody data, immunity data, because there is a correlate of protection, and because meningococcal infections are so rare, if you would want to see the difference between infections between a vaccine group and a placebo group, you would need trials larger than, well, hundreds of thousands of individuals, and it is logistically and financially not feasible. So that's why a phase 3 study is actually, for meningococcal vaccines, is actually a large phase 2 trial. And then for COVID-19 vaccine trials, most of the trials were between 20 and 40,000 participants, which are large studies. And that is based on power sample size calculation. What is the attack rate of the virus? What is the expected difference or efficacy of the vaccine as compared to placebo? And then another exceptional situation was a situation of rotavirus vaccine development. These trials were exceptionally large, 60 to 70,000 babies. That was because the first rotavirus vaccine actually induced a very rare and severe side effect, intussusception. So it was an intestinal complication. And because of that, the first rotavirus vaccine was actually withdrawn from the market. And the next vaccines being developed had to demonstrate that they did not induce this severe side effect. So therefore, these studies were larger than the ones we generally see. So how was time saved? How was the process accelerated? So you can see here what the standard process is of vaccine development. So first of all, the quality of the product that is being evaluated, followed by the preclinical studies and animal models, followed by the clinical studies, phase 1, 2, and 3, followed by the evaluation and authorization. Then the production starts, and then, of course, the post-licensure studies, the phase 4 study starts. How does this timeline look like? So it has already been mentioned as well that this regularly takes between 5 to 10 years. And this has been accelerated for COVID-19 vaccines. But as you see, the different steps are the same. None of these steps have been skipped. What is different is that these steps run in parallel. And production, for example, has already been started prior to the licensure of vaccine. Scientific evaluation is not done at the end of phase 3, but is done continuously. So when data are accumulated, when data are collected, they are sent in to the FDA and EMA for what we call rolling review. So summarized, the accelerated development process, or so how did we win that much time? Is by giving priority to COVID-19 development, and that's vaccine development, and it's clear why. It's a virus that dominates the whole world, so it is of highest priority. Huge budgets have been made available. Just one example of the American government, Operation Warp Speed, gave more than $10 billion for the development of 6 vaccines. But of course, other instances, the European Community, CEPI, the Coalition for Epidemic Preparedness Initiative, the Gates Foundation, so a lot of initiatives to support the rapid development of vaccines. Short review timelines. This is something we have never encountered before, but our ethics committee, for example, gave absolute priority to COVID trials and gave approval within 4 days. Some people found this a little bit frightening, saying, well, somebody cannot evaluate such a file in 4 days, but we already had to send in draft protocol versions and discuss already in advance what the study would look like and so on. So in a very early stage, regulatory agencies and ECs were involved. Phase 1 and 2 and phase 2 and 3 trials ran in parallel or actually together in one study design, which we call a seamless design. For example, the CureVac trial we are involved in, the Herald study, is a phase 2-3 trial, which means that it's one big study. The first 5,000 subjects were phase 2 participants with a more intense follow-up schedule of visits and blood samplings. And these data were sent in the federal agency and evaluated if there were no safety concern. And actually, after a couple of days, we could just start phase 3. Normally, in other situations, it takes months and even sometimes a year in between studies. So that was also a way to gain time. Some companies who had experience with the same platform, for example, Johnson, Johnson & Johnson had their adenoviral vector. They already had a safety database with more than 60,000 data from more than 60,000 individuals. They could use these data because it's actually the platform itself that accounts for most of the side effects. And the antigen, which is incorporated, has, of course, it needs to be evaluated, but the platform itself actually determines a large part of reactogenicity of safety. Because of the fact that side effects occur mainly shortly after vaccination, based on all the experience we have, regulators, both in the US and in Europe, defined upfront that safety data up to 8 weeks post-vaccination were sufficient for regulatory review. So we did not have to wait until the study was ended, until 1 year or 2 years for the data, for the file to be complete. If the vaccine was shown safe in individuals after 8 weeks post-vaccination, that was sufficient for approval. Of course, the follow-up of these individuals continues, but long-term follow-up shows us that there is no safety concern beyond that time of 2 months. I've already mentioned the rolling review, the large-scale production that started early, and these novel technologies, in particular the messenger RNA vaccines, which are which is a synthetic production process, have a much shorter production lead time. A traditional vaccine, for example, can take easily from 12 to 24 months, and the routine vaccines we use can take from 12 to 24 months for them to be produced. So this is a lot quicker thanks to this new technology. Some words on the CVAC experience, very briefly, and again, I apologize that it is in Dutch, but I will summarize. If you look up, if you Google us on the internet, which I did yesterday evening, you can actually follow our activities in the media. And that was also something which was completely new for us, because we're used to working under the radar, in silence, and working hard. In all the vaccine trials we have already done, and the media attention was huge. Every step that was taken was actually covered in our national media, starting last year in June with the CureVac phase 1 study. Then a month later, we started the second trial, a phase 1/2 study of Janssen, which was halted in October. We wanted to keep that quiet, but it was picked up very quickly by the media, saying that also the University of Ghent, University Hospital of Ghent, has put the J&J study on hold. In October last year, so 7 months ago, we did a call in the media asking for candidate participants to register. At that time, we expected to be able to enroll about 2,000 participants in phase 3 studies in our center, and we had more than 5,000 people who registered in our database. This is something we have never ever experienced before. In the end, we did not include 2,000 individuals, but around 1,000 to be precise, a little bit above 1,000, more or less. And that was mainly because of the fact that the start of the phase 3 studies encountered quite some delay. Companies were in general very optimistic, and even if a lot of time could be gained, it still is a lot of work to set up a large multi-center international phase 3 study. So the phase 2/3 study of CureVac started right before Christmas in our center, the Herald study, and 1 month later we started the Ensemble 2 study. And all of these studies are still ongoing. It sure is a fact that COVID-19 has boosted clinical research, research for novel drugs, antibodies, but also vaccine research. And again, from our personal experience, we had a full schedule with trials in 2020, and we were able to postpone some studies, but we also expanded our team very rapidly from 25 to 40 staff members. which we hired very rapidly and trained very intensively. And we noticed that our unit of about 450 square meters became too small. And especially in these times of social distancing, it was unacceptable to have our volunteers packed in waiting rooms very close to one another amidst a COVID crisis. That was, of course, unacceptable. So we got the support of our hospital direction, and we went looking in the neighborhood of our clinical unit and found this old warehouse of the electricity department, which was 8 meters high. A floor was actually put in the middle of this high warehouse. And in 3-4 months, and you can see here the floor, this is the upper part of this warehouse, and today this is how it looks like. So this was realized, this was actually constructed or refurbished in less than 4 months. The fact that you might hear some background noise is because my office is actually situated in this new unit, and people are still working and finishing the new unit, which is preparing us for the future. We have 8 extra consultation rooms, so we are ready to deal with future clinical trials. And another very impressive achievement is the construction that started of Vaccinopolis. Colleague Professor Pierre van Damme and the University of Antwerp, in collaboration with the University of Université Libre de Bruxelles. It's a public-private partnership with a lot of support of the federal government. It was planned to be finalized in 3-4 years, but because of COVID-19, this project has been also prioritized and accelerated. And this innovative center will actually have 30 beds for what we call CHIM studies, controlled human infection models, where healthy volunteers will be challenged with a virus, with a well-defined virus, and will be vaccinated before to see what happens and to see if a new vaccine might be effective, yes or no, and needs to be further developed. So what have we learned from clinical trials until today? The clinical trials that have been run have given us data on immune response, immunogenicity, and that's mainly phase 1 and 2 studies, on efficacy against laboratory confirmed COVID-19 infection. Those are the phase 3 studies. Until now, all of these studies have been placebo controlled, but it becomes difficult, and I will elaborate on that in a few minutes. And safety data are generated in all phases of clinical research. So just a quick word on vaccine-induced protection. So if we talk about vaccines that protect, it's always very important to ask the question, protect against what exactly? Against which clinical outcome? Against COVID-19, of course, against the disease. But there's various, we all know that the virus causes a spectrum of disease, ranging from mild forms of disease, even asymptomatic disease, moderate to severe disease, and can even cause death in some. So if we develop a vaccine, our primary aim is, of course, to prevent mortality and to prevent severe disease. And of course, it is very nice if we have a vaccine that also prevents the mild forms of the disease, and in the ideal world also prevents asymptomatic infection. Because if the vaccine can prevent asymptomatic infection, becoming infected without any symptoms, it also most likely also has an effect on transmission, and those are the vaccines that also can induce herd immunity, group immunity, or protection. When you look at papers, at results, at articles in the media, it's also very important to see if they talk about a vaccine that is protective, that much or that many percent, which virus caused the infections. Because of course the manufacturers that started early on and are already licensed, they were in a situation where, for example, Moderna, Pfizer, the majority of infections were still caused by the ancestral strain, the original Wuhan strain. Studies that started a little bit earlier, AstraZeneca already, J&J, and the ongoing clinical trials, the majority of infections was caused by other variants of concern, mainly the UK variant, the B117, but depending on the location in the world, other variants were predominant. And of course, there's a lot of concern right now on the B1617 variant or the Indian variant, whether the vaccines based on the Wuhan strain will protect or not. Coming back to these clinical outcomes, it is important to say that every company started that, I mean, the timelines were so short that there was no time to harmonize clinical trial protocols prior to the start of the study. So every manufacturer has actually defined themselves what the definition was for mild, moderate to severe infection, what the threshold was to take a sample, to take a swab, because of course you need to demonstrate the presence of the virus, and the trigger for sampling was different. So there's no standardization of clinical definitions, which makes it difficult to directly compare results across clinical trials. The gold standard for measurement of protection is still the placebo-controlled phase 3 clinical trial. But of course, in addition, since the vaccine is being used already at large scale, effectiveness data from real-life conditions, from epidemiological trials, although they have methodological shortcomings, they are very useful as well. How about immunogenicity data? I've already explained a little bit that we do know what is important and what we need to measure in blood, but as the immune correlate of protection is not completely defined yet, it is, as of today, not possible to replace placebo-controlled study with an immunogenicity trial. Viral analysis data are also very important when a vaccinated individual becomes infected, to have sequencing data on the virus causing the infection, to know what could be if it's an escape variant against which the virus is not working, for example. And then the challenge studies, which I mentioned in the context of the Vaccinopolis project, but also non-human primate studies in chimps, for example, are providing a lot of useful information. So it's the combination of all of these bits and pieces that gives us an idea on how protective these vaccines are. But again, if it's not within one clinical trial or within one epidemiological trial with the same definitions, with the same laboratory tests, within the same epidemiological context and the same study population, please do not directly compare and say this vaccine is 25% more effective than the other vaccine. It's really comparing apples and pears. Phase 3 trials are a big endeavor, as well as the other trials, of course, and every company has used its international network to conduct these trials, and it's always very useful and valuable that a study is not being conducted in one part of the world, because of all the reasons I mentioned: ethnicity of the study population, underlying risk factors and chronic diseases, the circulating viruses. This is just one example of the Oxford AstraZeneca vaccine, which is actually they started off much more. How do you say, it's actually the vaccine developed by the Oxford University. So that the project really started as an academic project, much smaller, with various smaller clinical trials. So the ones you see that started in April, May and June were sponsored by the University of Oxford and not by AstraZeneca at that time, the one in South Africa, for example. And it's only later on that AstraZeneca took over, and the later clinical trials, so the big ones here, 30,000 individuals, was sponsored by AstraZeneca. So the fact, I think a lot of lessons can be learned and will be learned from this pandemic. But the fact that various smaller clinical trials with various definitions were set up and with various intervals made it very hard to actually pool these data and have one good point estimate on efficacy. And that actually, I think this has led to the more difficult road of development that the University of Oxford, in collaboration with AstraZeneca, has encountered in comparison to other companies. So to summarize, the protective, how protective are these vaccines? Well, they're very protective. between 65 and 95% against all forms of disease. And if you look at severe disease, which is of course the main endpoint, protection rate is even higher. And these data are derived from phase 3 clinical trials and real-life effectiveness data. However, the majority of these cases were still caused by the Wuhan strain and the UK strain. Data are coming out on protective efficacy, effectiveness against variants of concern. Of course, time does not allow us, and it's not of all the purpose of this talk to go into detail on each of the individual trials and the results thereof. But important thing is that the vaccine has a good or quite similar vaccine efficacy effectiveness against the UK strain. Has a lower vaccine efficacy or effectiveness against the other variants of concern, but until now it seems to be good against severe COVID-19. And let's hope that it remains that way until the new versions, the adapted versions of the vaccines are being developed. It has been decided that the South African variant, the B.1.3.1.5.1, should be the variant to be used by manufacturers to make or develop a new version of their vaccines. The fact that the vaccines are still protecting against severe COVID-19 demonstrate or is indicative of the fact that the cell-mediated immunity induced by these vaccines still accounts for this protection, because cell-mediated immunity is less susceptible to the mutations occurring. These epitopes are much more stable as compared to the antibody-mediated immunity. How safe are the currently available vaccines? The fact that they are licensed demonstrates that they have a very good safety profile. Of course, it is a medical intervention, so there always are side effects, and a risk of 0 does not exist. With none of the medical interventions are completely risk-free. So the most common side effects are the ones depicted here, due to the fact that the immune system is induced. And actually, some people say you shouldn't call them side effects. They're actually a sign of the effect of the vaccine. It is normal that these symptoms occur and arise. They're self-limiting. They're not. They can be annoying, of course, but you can explain them. And it's actually what we expect, especially in young, healthy people with a very vigorous immune system. This is more or less what can be expected. It's not because you don't have these symptoms that you don't have a good immune response. And that's also something we know. Symptoms decrease with age, and that is, of course, due to the aging of the immune system. Something we were worried about, a lot of people were worried about prior to the start of development of the vaccine, is a phenomenon called vaccine-associated enhanced respiratory disease, which has been seen especially in animal models with other coronavirus vaccines, being that a vaccinated animal or vaccinated individual that encounters the virus becomes more seriously ill than when there is no vaccination. And already from the phase 1 studies onwards, competent authorities have required a very good surveillance. So already in phase 1, we had to take swabs and document COVID infections to really prove that there is no enhanced disease. And in the meanwhile, there is reassuring and a lot of evidence that this is not the case. Of course, in the meanwhile, we have a lot of real life experience, which provides us larger data sets and more data on safety. So what will ongoing and future clinical trials learn us? First of all of the licensed vaccines that have been approved by EMA received what we call conditional marketing authorization. And I just took this print screen from the Pfizer-BioNTech vaccine, but it's the same for the other ones, saying that the vaccine receives conditional marketing authorization. That means that the company has to continue to provide results from the main trial, which is still ongoing. The trial and additional studies will provide information on how long protection lasts, how well the vaccine prevents severe COVID cases, because in the primary analysis and publication, there were not a lot of cases of severe COVID disease, how well it protects vulnerable groups such as immunocompromised people, children, and pregnant women, and whether it prevents asymptomatic cases. So these are studies that are ongoing in the post-marketing phase. So ongoing and future trials will give us information on what I just mentioned: safety, immunogenicity, efficacy in these specific groups, children and adolescents included. Important to say that it is not because studies are being done in children that it has already been defined that children, and in particular young children, need to be vaccinated. But in any case, we want to be prepared in case we notice that the epidemiology shifts toward younger age groups and children become an important driver of the epidemic. In that case, it becomes important to vaccinate them. Persistence of immune response, and then of course the question, do we need booster vaccines, and how will the response be on these booster vaccines? A very important trial started, I think yesterday or the day before in the UK, the cough boost trial. Prevention of asymptomatic infection is being evaluated in many different studies by having swabs taken by all volunteers independent of experiencing symptoms. And then there's also a lot of interest in mixed schedules, in dose sparing. For example, Moderna uses 100 microgram of mRNA. Do we really need 100 micrograms? So there's studies ongoing, and already some have been published on 50 microgram. For example, alternative intervals that allow more flexibility. So there's a study in Oxford. I think the first results are being released now. And in Belgium, we are starting a study next week, sponsored by the Federal Knowledge Center and in collaboration with colleagues of Antwerp, of Brussels, which will also look at the dose-sparing capacity of an intradermal injection of the vaccines. So injecting only 1/5 of the standard dose of a vaccine. So we're very curious to see what the results will be. Co-administration with other vaccines is something which is normally evaluated in the course of the development process of these 5 to 10 years. This is also something which is being explored now in the later phase. And then very important trials is, of course, these adapted version of the vaccines, the 2.0, how there's what we sometimes call them, with the variant strain, South African or another one. And of course, the companies that are a little bit later in the race, how do they have to evaluate the efficacy of their vaccine? Do we have an alternative trial design for the placebo-controlled phase 3 study? Because that becomes really a problem. And it's, of course, we know that a lot of people still need to be vaccinated. So we need to stimulate companies to pursue their development and create, and a new framework needs to be created for phase 3 studies. And this is an endeavor CEPI, amongst other, and COVAX is undertaking, and I took just 2 slides from a recent workshop, a COVAX workshop, explaining that it becomes increasingly difficult to perform placebo-controlled trials because we want to have high-risk individuals in studies, but these high-risk individuals, in particular in the US, in European countries, these high-risk individuals are already being vaccinated. So they will not agree to step into a placebo-controlled trial. Also, younger groups, they will be vaccinated in the coming months, but it becomes more and more difficult to stimulate them to step into a placebo-controlled trial for and wait with their vaccination for another 6 months or longer. And even in countries with the limited vaccine supply, enrollment of volunteers, of healthy young volunteers, has also slowed down, and this extends the recruitment time and makes it even more difficult. So a lot of dropouts. And it's something we can confirm. It's also from our experience that there's an impact on data quality. For example, if a volunteer experiences no local reaction after the first or second vaccination, he or she, concludes that they received a placebo and will seek vaccination elsewhere. We even have volunteers that go to their general practitioner to have their anti-spike antibodies determined, to know whether or not they have received the vaccine. This is all very exceptional situations. I mean, We understand, of course, but this does have an important effect on the blinding, on the design of the study, and on data quality. The majority of the trials have been done in seronegative individuals or individuals without a documented infection, and it becomes also more difficult to find these. So what will be the next type or an alternative study design? It's also maybe a busy slide, but just summarizing the essence of this slide. So the default situation, or the ideal situation was, of course, the ones Moderna, Pfizer, AstraZeneca, J&J were in. They had this prototype vaccine based on the original strain, and they could do a conventional placebo-controlled efficacy study. But for a study like CureVac, which is ongoing, it becomes very difficult, as 1 in 3 individuals, at least in our center, has already asked for unblinding. So, the new situation, or the companies that have to start their trials either with an adapted version. So let's say Pfizer or J&J wants to license their South African variants of their already licensed vaccine platform. Well, for those type of vaccines, a non-inferiority study based on immunobridging will most probably be sufficient. So within one study, there will be one group receiving the Wuhan vaccine, another one receiving the South African variant, and then antibody titers will be compared between both and have to be non-inferior to the ones induced by the original vaccine. For companies that are not that far and have no licensed vaccine yet, they will have to compare within one study with a vaccine of a comparable platform, if available, another mRNA vaccine, another adenoviral vector vaccine, and so on. We do not think that a lot of companies will start phase 3 trials still with the Wuhan strain. So it is recommended that starting phase 3 studies should include already a variant of concern. So to end, what will real-life experience tell us? I've already told you, there's a lot of real-life data from epidemiological data, viral sequencing data. These are the type of studies or data sets, big data sets, that will tell us something on the rare side effects, on safety in populations excluded from clinical trials, the impact of the variants of concern, and the impact on overall epidemiology. And it's very reassuring to see graphs like this one, which I found on the website of BBC, showing that countries which are more advanced in their vaccination rollout, in their vaccination campaign, such as Israel, UK, and US, already see a rapid decline of the number of cases, even in groups that have not been vaccinated yet, so showing already some degree of herd immunity, whereas other countries are now coming into that phase. And I hope everybody will have, all countries will have rapid access to vaccine. Time does not allow me, because time goes very quickly once you start talking, but the vaccine-induced thrombosis with thrombocytopenia observed after vaccination with AstraZeneca, but also with the other adenoviral vector vaccine, is an example of a safety signal that has been picked up in the post-licensure phase, and it is too rare to be observed in clinical trials, and that is independent of COVID or not. This is just too rare to be observed, and an example of this risk-benefit analysis. And based on this analysis, it has been decided in Belgium that the vaccine Vaxzevria is not recommended below the age of 40 years, because the number of, in this case, ICU admissions prevented is not that high as compared to older age groups, and the number of these cases of blood clots with low platelets is quite substantial in this age group. So to conclude, I think expertise, sufficient financial means, and very hard work, teamwork, have led to the incredible achievement of having 4 EMA licensed vaccines and other vaccines, of course, in the rest of the world within 15 months after declaration of the pandemic. COVID-19 pandemic has been an opportunity as well for acceleration of the development of very successful novel vaccine platforms. But we need more vaccines because a lot of people around the world have not been vaccinated, and there is a large and unacceptable inequity of access to vaccines. Clinical research has generated a lot of knowledge and insights and will remain important the coming years. And I would like to end by saying that without volunteers, there's no new drugs, also no new vaccines. So a big thank you to all of the study participants in our center, other centers, and all over the world. Thank you very much.
Well, thank you very much, Isabel. Very nice presentation. Very a lot of information, and I'm sure people will have to digest this. We do have a couple of minutes left for question and answers, and yeah, I see already signs of appreciation from the audience, which is always great. Maybe, well, we'll probably not be able. They ask questions in the chat. We'll probably not be able to like address them all. There's a couple around like the special populations. Do you know anything around pediatric populations? What is the status there? Have they been tested, like pregnant or lactating females? Can you like elaborate a little bit on that?
The first study I saw, at least this week, was published in JAMA, if I'm not mistaken, a small study, a convenient samples or a small study in pregnant women showing an immunogenicity which is comparable to a non-pregnant population and showing no impact on the outcome of the pregnancy, so reassuring data. So there's clinical trials ongoing. That was just a small one, but companies are also performing these studies. I have not seen, but it's hard to keep track of all the published studies. Actually, you would need somebody beside you. Just, I mean, the number of studies being published is just incredible. But there's studies ongoing. We don't have any reason to think that there might be a risk. So that's why, for example, the Belgian Superior Health Council, the NITAC in Belgium, has already recommended the standard vaccination of pregnant women, even before these large-scale studies being published, because it's a theoretical risk. It's not a live-attenuated vaccines. None of the vaccines used are live-attenuated. So it's about a theoretical risk. And on the contrary, you see that even healthy women, pregnant women without any risk factors, end up in the ICU. There have been even fatal cases. So that's why we in Belgium started recommending it to all pregnant women and not only to high-risk pregnant women or healthcare workers who are pregnant. But studies are coming out and are being conducted, maternal immunization studies, yes.
Excellent. And for the pediatric populations, maybe question in general, like which age groups and how is this done? Is it with like children like below 12 or lower or 5 or lower? What's typically the standard?
So Pfizer was the only company who started evaluating the vaccine from age of 16 onwards. So that vaccine is already licensed for 16 and 17-year-olds. But other companies, including Pfizer, are doing studies in adolescents first, so 12 to 17 years, and planning and already started, if I'm not mistaken, studies from the group from 6 to 12 years. I'm not aware of any company evaluating safety and immunogenicity in younger children, in children below the age of 6 years. Also because the epidemiology, there's not a lot of serious infections in that age group. So right now that is not a focus. These are data focused on safety and on dose finding, especially in younger children. The question is, do we need that dose or can we reduce the dose also in order maybe to reduce side effects and to spare antigen? And those are not placebo-controlled efficacy studies, but non-inferiority studies compared to the immune responses in adults.
Right. Okay, thank you. And are you aware of any studies on the transmission of the virus and the impact of the vaccination on that?
Yeah, it's a good question. Well, there are studies on both clinical trials and epidemiological data on asymptomatic infections. So having swabs taken by vaccinated individuals at a regular basis and comparing that to non-vaccinated. So we know increasing evidence that if you're vaccinated, the risk of getting infected asymptomatically is lower. It depends a bit on study, from one study to another, between 30, 50%, depending. And if somebody becomes positive, a positive PCR, despite being vaccinated, the viral load is lower as compared to a non-vaccinated group, and the duration of viral shedding is lower. So therefore, it is concluded that the transmission might also be lower as a consequence. But in order to have really transmission studies, that is also being undertaken in various parts of the world, in Brazil, amongst others, the setup is a trial in which you vaccinate just one family member, the other members of the family are not vaccinated, and then you do follow-up with swabbing to see if the vaccinated individual becomes infected, if there's transmission, in which direction there is transmission. But these studies, as far as I know, have not been published. Quite complex, yes. But we use asymptomatic infection with positive PCR and viral load as an indicator of infectiousness and transmission.
Yeah, understand. Maybe one more final question, because I don't want to go too much over time. There's still some people expressing concerns about the speed of the development and the research being done. How do you, and I think you alluded to it already in your presentation, that actually the safety concern is mainly for after vaccinations after up to 6 weeks. But how about long-term safety? There's still concern in the chat on that.
Yes, the fact is that if you tell people that the normal development of a vaccine takes 5 to 10 years, people think that we follow up people that long, because we might think that, and I always ask people, are you aware of any long-term side effect of any of the more than 30 licensed vaccines? I mean, there's a lot of, in some countries, there's excellent surveillance systems with the health databases and where you could really monitor after the introduction of a new vaccine, for example, HPV vaccine in a healthy population. They monitor the occurrence of new onset chronic diseases, autoimmune diseases, and so on. This is something which is very carefully monitored. And of course, it can always happen. It's if you vaccinate a large population, things will happen. People will die shortly after vaccination. They will develop diseases, but it is very important to look at the background frequency of that event and see if this specific event is occurring at a higher frequency than what we expect. And that is what has been seen for this VITT, for example. So showing that the VITT cases all occurred between 4 days and 4 weeks after vaccination.
Right. Well, thank you very much. I think, and I think, well, there's more questions, but we possibly can't deal with all the questions. Because for the sake of time, because I see that people leave, and I really want to wrap it up properly. In the first place, by thanking you, Isabel, very, very appreciated your valued contribution to this this information session. I have seen a lot of signs of appreciation also in the chat from the audience. So thank you for that, for being present with us. Also, thank you to the audience for being here. You have our contact details on the screen. We will be sending you a short survey, and we will also, after completing the survey, we'll be happy to send you the recording and the materials, so you can also listen in afterwards. So with that, I'd like to thank you all again, and hopefully see you again in the next opportunity. Thank you very much.